Welcome everybody to my PhD defense. Welcome to jury members and to the reviewers that I would like to kindly thank for the work and the time they spent on my PhD thesis. So my PhD thesis is entitled "The Paradigm-Free Regularization for fMRI Brain Activation Recovery." The thesis director is Dr. Rachid Derish, and the co-advisor is Samuel Delaurier-Gauthier. In this introduction, in this presentation, I will introduce the problem in the context of the project. I will give a background. I would talk about the state of the art. I will introduce and describe our temporal regularized deconvolution of the fMRI Bolt signal. I will then describe our paradigm-free functional MRI approach and then give a general conclusion. The main topic of the project of this thesis is the human brain function. And when we talk about a human brain function, we refer to the ability of the brain to perform a physiological, a motor, or a cognitive function. And the brain does these at different scales. So on one hand, a brain function is achieved by the cooperation of neurons that are close to each other, that are located in a specific brain regions. And in this case, we refer to the concept of specialization. On the other hand, the neurons are connected via white matter fiber bundles, and so there is an association of neurons that can communicate even if they are located in segregated regions. So there is a dynamics going on in the brain, and it is a crucial point to provide tools to allow a comprehensive brain study. The neurons, they communicate via conventional signals, electrical signals, and neuroimaging techniques. They try to fill in the gap between how the neurons work and what we see in practice, in practical life. Neuroimaging techniques are non-invasive. They provide several in vivo measures in the brain. And they do this at different resolutions. Neuroimaging techniques, though, they give measure the tests that are degraded. So there is the need for techniques that allows us to extrapolate some meaningful information about the brain functioning. In this project, we were focused on the functional MRI that gives an indirect measure of the brain activity by measuring the metabolic activity that reflects neural activation. It has a high spatial resolution of the order of millimeters, despite a low temporal resolution on the order of hundreds of milliseconds and seconds. So in this project, the problem we are dealing with is to know where the brain activates, for how long it activates, and when it activates and deactivates. So we want also to know this, to do this without the knowledge of the task. Traditional techniques, they are based on experimental paradigms where the subject is asked to perform a task, and these provide us with some difference that we can measure between a baseline and a task condition. So in traditional techniques, this task, they gives us hints about where to look for an activation and when to expect an activation. But what about those subjects who cannot perform a task? This is important nowadays with the increasing of the life expectancy that provokes the fact that there is a higher impact of neurological diseases. And what about also those subjects whose activity does not follow a stimulus, like, for example, in the case of epilepsy? So, interestingly, there are techniques that allows us to measure the brain activity while the subject is at rest. So he's not asked to perform any task, and so in the absence of a stimuli, and this emphasizes the need to recover the underlying neural activations without the knowledge of an experimental paradigm. So our challenge, our goal is to approaches for the recovery of brain functional activations without a prior information on their timing, their duration, and location. So the fMRI technique is based on the bold contrast. So subsequently to a neural activation, there is a vascular response. So there is a change in the ratio between the oxygenated and the deoxygenated hemoglobin that are considered, that are exploited as endogenous contrast agents. So this change in this ratio provokes a change in the signal that we measure, and so we can. And so this is the bold effect. The bold response to a brief stimulus can be represented with a hemodynamic response, and so we can see that there is a peak in the signal that is measured after 5 seconds, and there is a response that lasts for about 20 seconds. There are two techniques for fMRI. We have on one side the task-fMRI, where the subject is asked to perform a task, and this provides us with an experimental paradigm that we can exploit when we are looking for brain activations. On the other hand, there is the resting state fMRI, where the subject is asked just to lay in the scanner, not to think about anything in particular, and so we don't have anymore this reference. In this slide, I show you how functional MRI images looks like. So we have 3D brain volumes that are acquired over time, and this results in a 4D structure of these images. So there is a 3D spatial dimension and a one-dimensional in time. fMRI images are corrupted by several sources of noise, among them physiological noise, head motion, thermal noise. And if data preprocessing, an appropriate data preprocessing is applied on the data, the residual errors can be assumed as white noise. There is also a big variability in the hemodynamic response across subject and within the same subject that is acquired in different scanning sessions. There are two, so let's now move to the state-of-the-art approaches. So there are two main groups of approaches for the analysis of fMRI data. So on one hand, we have the signal recovery methods that exploit to recover some information related to the signal of each voxel of the brain, and they include the hemodynamic model, among them the GLM and the deconvolution approaches. On the other hand, the connectivity-based approaches aims at group voxels that show some kind of similarity in their time courses. In these approaches, they do not include any hemodynamic model. So the GLM is a traditional technique, And in the GLM, each voxel time course Y is represented as a linear combination of regressors weighted by some parameter beta plus an error term. And solving this GLM problem corresponds to estimate the beta values that minimizes the residuals. And these beta values, they tell us how much that voxels contributed to the given task. This approach requires, though, the offset of the experimental paradigm that are the so-called regressors. And so it is exploitable for task-fMRI, but it's not exploitable for resting state fMRI. We have also the deconvolution approaches. So the deconvolution aims at reversing the effect of a convolution that explain the acquired data. This is done by Fourier transform in absence of noise, but fMRI measurements, they are corrupted by noise. So if we have an observation model given by the linear mapping that I show here, this is usually solved by minimizing the residuals, but given the presence of the operator H, this is an ill-posed problem. So additional constraints are required to constrain the set of solutions, and this is what is called the regularization. And these constraints are usually hypotheses on spatial and temporal features of the data. So among the convolution approaches, the total activation is a very widespread and recognized approach that implement the convolution approach. So they basically have a global problem where they include temporal regularization, where they minimize the total variation of the signal, and spatial regularization, where they exploit the fact that the activity inside a region is smooth, while the activity across region has crisp changes. They solve this problem by splitting this regularization problem into two regularization problems. So they have to set two different regularization parameters, one for the temporal and one for the spatial one, and then they combine the result to obtain a unique result by a weighted sum of the previous two. And this means that they have to choose to set other two parameters, omega t and omega s. As for the connectivity-based approaches, we have the seed-based approach, where that provides with a functional connectivity map that is obtained by computing the correlation between a region of interest and all the other voxels of the brain. We have also the clustering methods that group voxels that show a high level of similarity in their bold time courses, and among them the hierarchical clustering, k-mean and c-mean clustering. Another widespread approach for the analysis of fMRI data that is well recognized is the independent component analysis, that group voxels whose activity show similar patterns and provide with a set of spatially independent components that include the well-known resting state networks and spurious components. This approach is limited by the fact that it does not include a hemodynamic model, and the number of components is user dependent. So let's now move to the first main part of this presentation. In this part we exploit the temporal feature of the fMRI signal, and we treat all the voxels of the brain separately, independently. So we have as forward model for the fMRI an activity-inducing signal that is a piecewise constant activation. So we convolve it with an hemodynamic response, and we add noise to this data. So this is our forward model. And the goal is to, our goal is to recover the fMRI brain activation U of t, given the acquired data Y of t. And we do this exploiting the fact that the derivative of the activity-inducing signal U of t is sparse, as it was proposed by Karanoglu and colleagues. So we propose a LASSO optimization problem, where we have a data fitting term, and we penalize the sum of the absolute values of the so-called innovation signal S. In the data fitting term, we have y minus x, and so the signal S is convolved with the hemodynamic response function, and here we use the one obtained from the one proposed by Princeton and colleagues, and linearized by Khalidov and colleagues to be exploited in linear convolution. We also introduce another operator that we call the alpha filter, and this links the innovation signal to the activity-inducing signal. So the interesting point of this filter is that it has a shape. So it's an exponential accumulation function, and it has a shape that can be adjusted by tuning a parameter alpha. So the higher the alpha, the sharp is the function. The lower the alpha, the smoother the function. And we introduced this accumulation function instead of a finite difference or the inverse of a finite difference, because it allows us to account for the noise that is still present on the data. So we want to smooth the data, but we want to keep the solution as close as possible to a piecewise constant activation. So finding a alpha that gives us a trade-off between these two intentions is a point that we have to solve here. So we solve the LASSO problem with the least-angle regression algorithm that outputs all for a set of lambda the solutions. So given that we have all important lambdas, we picked the optimal solution using a L curve. We picked the optimal lambda and its associated solution using L curve. So to validate this approach, we firstly validate the way of the optimal lambda selection. So we compared the the L curve for the selection of the optimal lambda with the leave one out cross validation. So here I show some results. So in green we have the simulated fMRI data. In black we have the ground truth, that is the simulated activation that we want to recover. In the red we have the solution provided by the L curve, and in blue we have the one provided by cross validation. And here we compute the error between the solution and the ground truth. And what we show is that the leave one out cross validation is more sensible to noise, and that the value of alpha equal to 0.75, the error stabilizes to a value. So this is the alpha value that we're going to use for the subsequent analysis. So we simulated the phantom fMRI data, starting from activation map. This is an auditory activation map, and we did this using the FSL POSSUM tool. We multiplied each activation value with two kind of activations. And this provided us with a 4D fMRI data structure. So each of these time series was corrupted by what we call the block type noise. And this aims at simulating false activations that still are observed in the data after the pre-processing, as was shown by Power and colleagues. After that, we added what we call model noise to simulate a random intrinsic electrical fluctuation within the neurons. We convolved with the HRF, and then we added noise to simulate the acquired fMRI data Y. So the results on the phantom data, we firstly show that we are able to distinguish between a voxel that is activated and a voxel that instead is not. So as we can see in the plot, again, the green is the fMRI acquired signal, the black is the ground truth, and the red is our solution. And this color code, I'm going to use it for all the theses. We can see that we are able to discern, to distinguish between an activation and a not activation. We compared our results with the one obtained by using the total activation approach that we used, and we used the one that is provided in the on the web that implements the total activation, and for the moment we discard the spatial regularization to be able to compare the results. It has to be stressed that the tool was used as it comes, and we didn't have to set any parameter for them. So we show that we recover lower errors between the recovered activations and the ground truth, and lower standard deviation if compared to the ones obtained with the total activation. And here is an example of a recovered time series, and we see that in red, that is our solution, we recover an amplitude that is very well fitting the ground truth, whereas the solution provided with the total activation in blue is underestimating the amplitude of the signal. So it is true that if we would compute the the correlation between these two signals, we would have similar results. But if we would threshold the recovered activation, using our approach would allow us to choose a higher threshold and then not include false activations in the recovered activations. We validated this approach also on real fMRI data. So we took one subject performing a motor task from the HCP database, and the signal we are seeing here are average in a region of interest located in a brain region that is activating during the task. In this case, is a tongue task. So we can see also in real data that we are able to find a solution that clearly show that there is an activation with respect to when there is not, and that this solution is well overlapping with the two gray areas that represent the time points where the subject is supposed to do the task. We also observed, though, that we recover two different kinds of activations for the same task, and in this case, it's quite longer than the task. And this could be done to the fact that there is a huge variability in the hemodynamic responses in subjects. So to conclude this part, we proposed a temporal regularized deconvolution of the fMRI Bolt signal, where we use jointly the LARS algorithm and the L-curve that allowed us to not to have to select a priori a regularization parameter, as it was the case for the TA. We added model noise and blocks of false activations in the simulated data. We validated our approach on synthetic and on real data, and compared this approach with the total activation approach, showing lower computational times and better results. Though LARS cannot handle a whole fMRI image, because the problem becomes too heavy to be solved in terms of computation. This work was presented in different conferences. It was presented at the MBC, at CoBCoM, and at CauCA. So let's now pass to the second main part of this presentation, where given the excellent spatial resolution of the fMRI, we exploit for the regularization both the temporal and the spatial features of these images. So the idea is that large image variations, they should be preserved because they occur if there is a brain activation or if there is an edge. Whereas small variations, they should be removed to reduce the noise. And to do this, we propose to use a 4-dimensional structure tensor of the image and partial differential equations to iteratively and anisotropically regularize the 4-dimensional fMRI image to enhance coherent structures of the image and not include new structures in it. So to do this, we exploit the link between the diffusion of physical processes and the image processing. So we can observe on the left an image of an isotropic diffusion, and on the right, an example of an anisotropic one. So the idea is to apply this to fMRI images. So what we propose is that we update the fMRI image at each iteration. And in this term, we have a data fitting term that prevents the results to be too far from the original ones. We have a regularization term, and we have a regularization parameter. So to explain the regularization term, we start by defining the structure tensor of the image. And the egg decomposition of this structure tensor gives a set of eigenvectors that explain the distribution and the orientation of the gradient of the image in a neighborhood, and their associated eigenvalues. So this allows us to distinguish between isotropic diffusion and an anisotropic diffusion, where we have a principal orientation of this image variation. And it has to be stressed again that this is applied on 4 dimensions, whereas here I represented 3 dimensions for obvious reasons. And so starting from this structure tensor D, we recomputed D tilde, that is the one that we employ here. So in this D tilde, we aim at reversing the effect of D, such that we reassign values to the eigenvalues. So lambda tilde 1 is set according to a Gaussian function, and the other 3 eigenvalues are set to 1. What this means that if lambda 1 is big, we want, it means that in the associated direction there is a big variation of the image. So we want to save it. We want not to smooth in that direction. So the lambda 1 tilde will be small. And this would lead us to an anisotropic smoothing. On the contrary, if lambda 1 is small, it means there is not a big variation on that direction. So we want to smooth, and we set lambda 1 as big as the other eigenvalues. So we have isotropic diffusion. And here we show what we mean. This is an example. So basically, if we have an edge, we don't want to smooth in the direction that is perpendicular to this edge. So we have a structure tensor that looks like this. On the other case, where there are not image variations, we have isotropic tensors. We validated this approach on phantom fMRI data. So again, we have an activation map that we multiplied each activation value for a piecewise constant activation. In this case, we didn't add blocks of false activation. And we did this for different amount of noises, and this led to different SNRs that are decreasing from the top to the bottom. So we observe a common trend. And if we zoom in one of these simulations, so if this is the simulated fMRI image, this is the ground truth we aim at recovering. This is our solution, and this is the solution provided with TA. So in this case, for TA, we used the the temporal and the spatial regularization. Again, we use the toolbox that they provide. And so what we show is that we are able to recover an activation that is well overlapping the ground truth if compared to TA, though we have a smoother solution. If we look at the errors between the simulated activation and the recovered signals, we show lower error with our approach in red compared to TA. And if we look at the person correlations, we show higher correlations using our approach compared with TA. And we also show that we are more stable to noise. So here I show an example of for one subject, I saw an example of the structured tensors that we obtain in real data, and I wanted to show how the structured tensors look like anisotropic tensors or isotropic ones. And in this graph, we report a qualitative comparison between the GLM and the PFF-MRI. So for the GLM in blue light blue, we report the beta values. So they gives us how much that voxel contributed to the given task, and the tasks are reported in the lines of the graph. Whereas in red yellow on the right column, I report the voxel-wise correlation maps between the recovered solution using PFF-MRI and the given task that was simulated in this case with the ones where the subject is performing the task and zeros elsewhere. I make this comparison, and what we can observe is that the regions that are activating for the different tasks using the GLM and the PFF-MRI are very well overlapped. And if with the GLM we find this region with the knowledge of the experimental paradigm, with PFF-MRI we find these regions instead without any knowledge on the experimental paradigm. We then compared the PFF-MRI with the total activation approach. And so here I show for one subject the recovered activation in a region of interest that is supposed to be active doing the considered task. In this case is task related to the tongue that we can see in the gray areas, and that we are able to distinguish between a region of interest that is activated and region that is not, and that we show higher correlation with respect to TA for the activated regions and comparable correlation for the not activated regions. Here I show the results for a group study. So these are the for each task. In red, we see the average correlation values between the correlation between the task and the recovered activation for different iterations using the PFF-MRI. And in blue, I report the results obtained with the TA. And so we show that from the first iteration, we show higher correlation if compared to TA, and we also show that we converge to a stable solution after, on average, 20 iterations for the hands and 30 for the tongue, and that 20 for the foot, and so on. So here, I report the results for. So here we have the spatial maps. Of the activations in time, and we can observe that we can see a visual activation right before the activation of the hand, and this is coherent because the subjects follow a visual cue before starting the task. We also applied the PFFMRI to resting state data. These are very big data with 1,200 time points, and we averaged the activation in a ROI located on the default mode network. And here we show the result for different iterations. In this plot, we show that we can observe, using our approach, a dynamic on the default mode network seen with the resting state fMRI data, and we can observe that not all the regions obtained using not all the regions composing the default mode network are active at the same time, but that there is a dynamics between these regions. So to conclude this part, we propose the PFFMRI approach that allows voxel-wise recovery of brain activations. It exploits all the 4D structure of the image at once without splitting it in its temporal and spatial dimension. It was the case with the TA. So we had to set just one parameter instead of 4. We do this without the knowledge on the experimental paradigm. This approach was applied to resting state fMRI and it improves the state of the art. It was also applied in a multimodal approach to locate the active regions of the brain and then filter the tractograms. This approach, though, doesn't include a strategy for the selection of the regularization parameter. So this work was accepted for publication. Its application was accepted for publication at GNE, was submitted to a TTTMI, was accepted for an oral presentation at ISMRM, was presented at OHBM, and had a power pitch at ICMRM as well. And we also provide a software contribution where we provide a Python package for the PFFMRI. So to conclude, our goal at the beginning was to provide tools for paradigm-free regularization for voxel-wise brain activation recovery. And to do this, we propose two approaches: a temporal regularized deconvolution of the fMRI data and the PFFMRI that exploits the whole 4D dataset. We demonstrated to improve the state-of-the-art approaches. So we overcame the necessity of having a prior information on the experimental paradigm, as it was the case for the GLM. And with the comparison with the TA, we showed results that were closer to the ground truth with lower computational time. We showed the potentials for the application to resting state fMRI data. That is our final goal. And yes, here we validated it on task-fMRI data because this provides us with the task paradigm, and so allows us to assess the results. But the final goal is the application to resting state fMRI. And we provide a tool to investigate the dynamics of the signal. As open problems and future perspectives, so we provide tools to that can be applied for the study of the dynamic of the resting state fMRI data. It could be applied to recover transients in the resting state networks. It could be exploited for future clinical applications. Also, the structural informations given by streamlines could be added to the problem to define a richer neighborhood. And also this could be applied to investigate the bold signal in the white matter fiber band, in the white matter that is a debated topic. So we, to conclude, we had 2 journal papers. We had several participations in conferences, among them an oral presentation and a power pitch at ISMRM. I also collaborated with other authors on other topics, and we have a software contribution for the PFFMRI. So I'd like to thank you all. I'd like to thank my supervisor and co-advisor. I'd like to thank all the members of the juries and the reviewers for the work they did on my thesis, Giulia and Marco Lorenzi for the CSI, all the Athena team, and also acknowledge the ERC grant. So thanks for your attention. I'm open to questions.